something on this year, and so anyone in our division will do it, and then I'll see if I can include you guys as well, because that seems a bit unfair. Right, okay, so Laura's taken almost every minute of this session, so I will squeeze mine into the remaining 8 minutes. But I work with Laura quite a lot, so although the work I do is quite different to Laura's. I'll start with my career path, because it is quite different. So I did a maths degree initially. Interestingly, when I was thinking about this, my A-levels I did to do, because I wanted to do medicine. That was my first thought, because I was always super interested in health and biology. And then I immediately realised, after speaking to someone who had gone on to do that at uni, that I could not deal with like blood and guts and things like that. And that was just not a viable career path for me. So I went on to do a maths degree because I really enjoyed that. And at the end of it, I remember my supervisor was like, you should come stay on and do a master's and do a PhD. And I just thought, I want to go and do something like actually useful, because I loved maths, absolutely loved it, but it seemed so abstract. We're just like solving little proofs all day, and I couldn't really see how it linked to real life. And if only someone had have told me about this back then, because ironically I went on to do what is arguably the least useful thing to mankind. So go to work in investment banking for the best part of a decade. So that is what I actually did straight out of my bachelor's degree. I went down to work in the city in London. I did love it. It was super fun. It was great. And I met some amazing people. And then I think I just kind of had this like quarter life crisis of like, no, you need to actually do something useful with your life, because that was always important to me. So I spent a good couple of years like really thinking about what to do, and it was quite serendipitous actually. It was just a meeting with a friend who was also a mathematician who was at UCL, who'd gone on to do a PhD in psychology. She said, you should do computational neuroscience. I did not know what the word neuroscience meant when she said that to me. I literally got my phone and Googled it, and I was like, oh yeah, that sounds really interesting. So long story short, I found a master's here in Manchester. There were hardly any back then. I think there are less now. I don't know how many computational neuroscience masters there are. There's one in Nottingham and maybe one in Plymouth. I'm not sure about any. There used to be one in Edinburgh. The one here doesn't run anymore. We obviously broke that. So anyway, I did that, and that was kind of my test. It was a year. It was the hardest year of my life. I think a master's is not for the weak-willed. It's extremely hard work, but I did absolutely love it. I met Laura during that master's, and so I went on to do my PhD with Laura, and we've been together ever since. Yes, bonded. So so the Medical Research Council funded the MSc, and then it's the Engineering and Physical Sciences was the PhD. I then went on to do research fellowship between here, Exeter, and the University of Oxford, and then went on to get, this is a bit old, this slide as well, and then in the end I became a lecturer here, so that's where I am now. So I'll probably just spend the time telling you a bit about computational neuroscience rather than going through any work, because I could come back another time, I suppose, if you're interested. Computational neuroscience, if anybody is interested in it, is actually a huge research field, and this is not the totality of it. This is my version of computational neuroscience. It includes so many different fields like advanced analysis methods, which we do kind of work on quite a few other things, but in terms of what I do, is much more focused on this intersection between kind of cellular and systems level biology, translational and experimental neuroscience, and mathematics. And then, of course, all the physics comes into it because I do quite a lot of neuroimaging as well. But, of course, this includes, AI and computer vision and all sorts of things like that. And you'll meet lots of people that say they're computational neuroscientists, but we do nothing compared, we don't really overlap at all. And I think the theme of my work, if I had to kind of sum it up, is really, I think that to tackle some of our greatest challenges is to kind of, we need to take multiple approaches across different scales, like temporal, spatial, because we tend to have this really super detailed, exquisite knowledge of what happens down here. Like we can describe cells, we can describe synapses, we can describe chemical reactions that go on in cells, all this kind of thing. But how does that? Often we want to know this. We want to know what's happening at the systems level. Why does Parkinson's affect movement? What's happening at the level of these cells that translates to these changes in behavior, or these macroscopic imaging measurements? Laura can show you these images of these changes in ASL in a tumor. But what does that mean? What actually is driving that? And that is what I think computational neuroscience brings to the table. It's a powerful, powerful tool that can allow us to integrate observations and knowledge across temporal spatial scales and provide this kind of integrated view from cell to system. So that's my kind of sales pitch. I think I honestly think it solves. It's in many instances it's the only tool that we have as far as I'm concerned. And it's kind of the life of a computational neuroscience goes a little bit like this. So, you can start anywhere you like. You can start with an experiment that either you do or somebody else does, and it will give you some data. And you can use that to inform a model, use that model to generate predictions. You use those predictions, they're testable. You test them with your experiment, and then you keep going, around and around. And we've done that, really successfully in quite a few instances now. So, I wasn't going to, I probably haven't got time to, because it is 1:45 now, so I was going to start talking to you about my actual research, but I think I could potentially come back another day, because I think it will take more than a couple of minutes. My actual research is focused on neuroplasticity. I can show you a couple of quick slides. Yeah, well, I can do if you like. You've got another 10 minutes. I can probably get through I was going to talk through a couple of experiments, so I can just talk through one.
We actually have 1 more hour for this room. So
okay. Okay, fine. Right. Okay, well, I'll go through one of the examples then. So you can get an idea of how we do it. I've tried to pick the most user-friendly one that you might like to hear about. Okay, so you're all in brain sciences, so you know what neuroplasticity is. It's the brain's ability to reorganize its structure, function, connections in response to some experience or learning or injury or growth or whatever. It's obviously this lifelong process. It underpins everything that you've ever learned how to do. And it helps us to learn new skills, build memories, and importantly, it's really important for recovery from injuries such as stroke. So my work, I have these kind of 3 pillars. So I work with humans and models, so I don't do animal work in general. And it's these 3 pillars. So how can we modulate plasticity in the brain in an optimal, targeted way? How can we measure plasticity in the brain? There's no answer to that. We can't, but we can have various proxy measures of how we might think we're measuring plasticity in the brain. And then how can we translate these findings to impact health? So I've got 3 of my project students in here. So they're working on one of the translational projects that kind of comes out of this work. But I'll talk about some work we've done here on how can we modulate plasticity in the brain. Okay, so non-invasive brain stimulation. These guys are experts in this now. You could probably give this talk for me. It's a very fast-growing and a promising area of research, and it's got great potential to offer treatments, therapeutics for diseases where we currently have very limited options. These techniques have the power to directly influence brain dynamics, and we think what they do is they're changing kind of connectivity patterns in the brain. And there's lots of clinicians and researchers using these methods all over in lots of different fields: stroke, autism, pain management. But despite all of the promise, anybody who works in this field will tell you that the outcomes are very variable because we don't know how to target the stimulation optimally. In most cases, apart from Parkinson's and epilepsy, probably. So, back to my original question, to be able to do this, we need to understand, what's happening at the level of the cell or the synapse that's kind of driving changes in behavior. And this is really an exemplary area of research where computational neuroscience can, enhance our understanding. Ooh. So this is quite old work, actually. I did this with Laura many years ago. I think this was part of my PhD. So our question was, can we optimize the frequency of sensory stimulation, so this is tactile stimulation delivered to the fingers, to promote cortical remapping? So to do that, we developed this computational model of a patch of cortical sheet. So each of these little squares is like a little neural mass. We relate it to a little patch on the cortex. I won't go into the maths of this, but essentially it's a specific kind of model called a neural mass model, where you have excitatory and inhibitory populations of neurons interacting with each other. And the connections between them are plastic. So that means they can change, they can get bigger and smaller. And then we stimulated this little patch of cortical sheet, and we systematically tested. I mean, it's quite simple. We just systematically tested lots of different frequencies, and that's the beauty of computational neuroscience. You can test thousands of different conditions, which obviously you cannot do in humans. And then, just the take-home message from that was that we had to use higher frequencies in order to drive this cortical remapping. So we wanted to test that in humans. And we decided to use the somatosensory system. So you're probably all quite aware of the somatosensory system. The reason we use it is because it has this very exquisite topographical mapping. It's highly plastic, and we do have full control of the sensory inputs. It's not like learning a task. That's much more complex, and you have this, so the organization of the input axons is maintained for the entirety of the tract, which is quite amazing. And so you've probably seen this, and so you end up with this mapping. So you know exactly which parts of the brain, it's topographically organized. Okay, so we just got these people in. We thought we could test this experimentally, and we just apply this stimulation to 2 digits on the right hand. We use a high and a low we just call them high and low. And we do some fMRI. And the first thing we found was that after the high frequency stimulation, these are the regions that code for the 2 fingers, had actually shifted across the cortical surface. That's after about 46 minutes of co-stimulation. And this shift, we hypothesize, is due to changes in connectivity between those 2 regions, essentially causing them to merge across the surface when they're activated. So was there any effect on behavior? That was another question we needed to ask. And again, we found the same effect. So this is a tactile, forced choice tactile discrimination test. I've said it so many times and I still forget how it is. Anyway, the basics of this is that you send a pulse to one finger or the other, and the people just have to tell you which finger it was. And then you stimulate it and for 20 minutes, and then you repeat the test, and you repeat the test, and you repeat the test. And what you can see is that the high frequency condition, these people are making more and more and more mistakes. It's just a linear effect. And the reason for that is the shifting of these digits across the cortex. So when they're apart and this one buzzes, it's quite obvious that this one buzzed. But when they're touching each other, it becomes much more difficult. So that was really interesting, but we wanted to understand a little bit more about the mechanisms underlying that. And what we thought at the time, because this work is quite old, but it's probably a lot more obvious now, was that inhibition must reduce in order for plasticity to kind of take effect. So we collected a kind of data called magnetic resonance spectroscopy. My students know all about this as well. And this is very similar. So you collect this in an MRI scanner, and it's really similar to all the things that Laura just talked about. The difference here is that we're not collecting the signal from water, the protons attached to water molecules. We're collecting the signal from protons attached to different molecular structures in the brain. So I won't go through this whole list, but it's basically an MRI scan. So it's non-invasive. You can quantify metabolites in vivo. You end up with a spectrum that looks something like this, and you can, choline, creatine, glutamate, NAA, you can measure GABA. The downsides of it is that you need a really, really big voxel. That's an actual voxel for an MRS acquisition. It's very, very big. We're talking about brain regions, not like, MRI voxels. But it's a really, amazing technique. And when you get a spectrum, so this is the output, it's not an image, it's this spectrum like this. And this, I stole this from, I should put the reference on here, it's from Bettina's paper, but essentially the y-axis tells you how much of each of these molecules, and the x-axis tells you what it is from where it is on the x-axis. So the two most interesting, we can measure a few different metabolites, we can't measure all of them, we can just measure some of the small ones. But two of the interesting ones, in my opinion, are GABA and glutamate. I think you probably all know what they are. These are the brain's major inhibitory and excitatory neurotransmitters. And so we repeated this, we put people in the scanner, we repeated this. And what we found was that, I think we found no change for glutamate, but we did find this reduction in GABA for the high frequency condition. So, it was really kind of, gave us this evidence that this repetitive stimulation at different frequencies can drive cortical remapping, but via GABA disinhibition. So I've got one little add-on to that, which I'll just keep going, and then I'll finish 2 more minutes. So the final add-on to this was, could we augment this effect by applying concurrent non-invasive electrical stimulation to the scalp? Just trying to put in as many techniques as I possibly can. So this is where we pass an electrical current through the head. These techniques are, they're low cost, they're portable, they're well tolerated, and they're really easy to administer. And these are the two kind of main ways you would use this. So you'd either apply a direct current that stays constant for the entire time, and this has been shown to modulate GABA levels as well, or we apply an amplitude modulated waveform which has the effect of entraining neural populations during stimulation. So it kind of has like a spike timing dependent plasticity effect. And so we did this, we just kept the frequency the same. So we just used this high frequency stimulation, but we had we had three conditions. One was sham. One was the two waveforms were applied in phase. So that's the waveform to the fingers and the waveform to the head. And the other where they were antiphase, so they were moving in opposite directions to each other. And there was some modelling work that went behind this, but I haven't got it today. It predicted exactly what we found, which was the sham condition. We just replicated our previous results. The antiphase condition abolished the effect, so they just cancelled each other out in the brain, and the in-phase tax kind of boosted the effect of the tactile stimulation. So yeah, that was kind of, we did have this augmenting of the effect by using the non-invasive brain stimulation. So I'll actually stop there. I'm not going to go through all three pillars today, but perhaps another time I can tell you about some of the others. Okay.
Wow.